Well, good morning, good afternoon, or good evening, ladies and gentlemen. Thank you for joining us for another video PowerPoint series for Unit 7, Genetics and Heredity. Today, what we're going to be talking about are the various types of dominance relationships that can be found with our Punnett squares and genetics in general. Specifically, we'll be talking about doing a dihybrid cross, discussing incomplete as well as codominance. So, without further ado, let's go ahead and get into this today, shall we? So recap, a genotype and phenotype. If you remember, your genotype is the combination of alleles. So if we go ahead and look here, right in the middle, our genotypes can either be homozygous dominant, recognized by two uppercase letters, heterozygous or hybrid, being one combination of an uppercase and one lowercase, so one dominant, one recessive, or we could have the homozygous recessive that shows us lowercase or two recessive alleles. The phenotype would be the physical characteristic those represent. So in this case, the dominant phenotype seems to be purple, whereas the recessive phenotype seems to be white. So if you show the dominant phenotype, what could your genotype be? So if you go ahead and show purple as your dominant phenotype, you could be heterozygous or homozygous. So you could either have two dominant alleles or one dominant and one recessive. And you would still go ahead and show the dominant phenotype. So if you're unsure if the organism is a pure breed, what you can do is something called a test cross. A test cross is where you go ahead and cross the dominant phenotype with a recessive individual. So what you do here is, if you're unsure if the individual that shows your dominant phenotype is a purebred or a hybrid, what you do is you cross it with a purebred recessive. If the offspring ends up being 100% dominant, the pure would have to be your homozygous dominant, because all of the offspring would be hybrids. If the offspring was not 100% dominant, so you get a 50-50, that shows that the original was heterozygous. If you go ahead and do the test cross here like we're showing, this illustrates exactly what we're talking about. So if we had our dominant genotype, dominant phenotype here, we want to know if it's either big P or big P little p. So homozygous dominant or heterozygous. The way you do this is if you cross it with a purebred recessive, in this case white. So if we go ahead and we get the results of all purple, all showing the dominant trait, then that without a doubt recognizes that we had a purebred dominant. Meanwhile, if we go ahead and do the cross and we result in a 50-50 ratio of dominant and recessive, that shows that you had a hybrid. So remember, Punnett squares are usually only good for single traits. So if we're just looking at one trait in particular, we call that a monohybrid cross. But what we can do is do something called a dihybrid cross, where we're looking at two different traits instead of one. Now initially these can look a little intimidating, but if you go ahead and go through this, you'll find it's actually pretty easy once you get down to it. So let's go ahead and try a dihybrid cross. Let's go ahead and show the probabilities of a cross between two people which are both heterozygous for full lips and clefted chin. Now clefted chin, that's the butt chin. You got a nice little crevice there in your chin. So let's go ahead and recognize that full lips is dominant to thin lips. So full lips being recognized as big F, thin lips being recognized as little f. And for the chin, we're going to use big B for cleft and little b for non-clefted or normal chin. Now the hardest thing to do here is actually going ahead and separating your combinations of alleles. So let's go ahead and go with big F, big B, little b, and big F, little f, that should be, sorry about that, big B, little b. Now the way we're going to go ahead and do this is using a little bit of the old math terms. Now hang on, don't check out on me yet just because I said math, hang in there. Trust me, if I can do this, just about anybody can. So, what we're going to do is utilize the math term FOIL. So, with math here, to go ahead and get our combination of alleles, you'll see that we have 4 by 4. We have a 4 by 4 grid. So to separate these, the first thing we're going to look at is the F, or first. So, for our first here, we have big F and big B. So you're looking at the first of both allele combinations. So we get big F, big B. Next, we go with the outer. So for the outer, we've got big F, little b. For our third, inner, we've got little f and big B. And finally, we have our last. So for our last, we're looking at the last 2 alleles in the combination. So that should give us little f, little b. Now luckily, since we already figured this out, and we're crossing 2 heterozygous individuals, so this and this are the same, we can just go ahead and copy what we put at the top onto the left-hand side. Now from here, it's relatively easy. All we're doing is just combining like terms, like we've done on every other Punnett square we've done to up to this point. So as you can see, we're taking the 2 big F's and putting them together. And we're also taking the 2 big B's and putting them together, which would give us full lips and a clefted chin. So let's go ahead and continue. For the first row, second column, we have big F, big B, little b, resulting in full lips and a clefted chin. For our third, we'd have big F, big B, little b, resulting in full lips and a clefted chin. For our fourth, we'd have big F, big B, little b, once again resulting in full lips and a clefted chin. For our second row, we've got big F, big B, little b, once again giving us full lips and a clefted chin. For our second, now this is where things change a little bit, we've got 2 big F's and 2 little b's. So we'd have big F, little b, giving us full lips but a normal chin. Our next one up, big F, big B, little b, resulting in full lips and a clefted chin. After that, our next one, big F, little b, giving us full lips and a normal chin. So there's another normal chin. For the third row, we've got big F, big B, little b, big B, giving us full lips and a clefted chin. After that, big F, big B, little b, full lips, cleft chin. Our third one, little f, big B, giving us thin lips and a clefted chin. So now we are showing the recessive for lips. Here we've got little f, big B, little b, giving us thin lips and a clefted chin once again. For our final row, we'd have big F, big B, little b, full lips and a clefted chin. Then we'd have big F, little b, giving us full lips and a normal chin. After that, little f, big B, little b, thin lips and a clefted chin. And finally, little f, little b. So we've got 2 of the recessive traits showing. Now let's go ahead and add up the combinations we have. Let's start off with what's going to give us the phenotype for full lip and clefted chin. So we have full lip, cleft chin, full lip, cleft chin. There's another, and another, and another, and one right here. Oh. Well, that just erased everything. So, not what I anticipated, but if you go ahead and add these up, which we'll do in the next slide, it'll show you exactly what we mean. So, we had, if you go ahead and look back at it, full lips and clefted chin, there should have been 9 out of the 16. For thin lip and clefted chin, that should have given us 3 out of 16. For full lip, non-cleft, once again 3 out of 16, and for thin lip, non-clefted, so recessive, should have given us 1 out of 16. So the ratio that you're always going to have here, when you're going ahead and crossing 2 heterozygous individuals for both traits, is a ratio of 9 to 3 to 3 to 1, being homozygous dominant, heterozygous for one trait, heterozygous for the other, and homozygous recessive for the last. So 9 to 3 to 3 to 1 is the classic ratio for a heterozygous dihybrid cross. So if they go ahead and give you 2 heterozygous individuals for a dihybrid, you actually don't even need to do the Punnett square anymore. Why? Because this ratio is always how it will turn out. Now once again, that's only if you're dealing with individuals who are heterozygous for both traits. So dominance relationships, we have a few of them that can vary a little bit. What we've been working with up to this point is complete dominance. One allele shows full dominance over the other, so it's dominant versus recessive. We can have incomplete dominance, where neither allele is fully dominant over the other. So we have a mixed phenotype. So it's kind of going to be like a combination of both. And our third type is codominance. With codominance, both alleles are dominant. So one phenotype wins in some cells and the other wins in others. So what we mean by that is it can show either. So for example, let's say we were crossing 2 flowers. One that was white, one that was red. If it was complete dominance, it's either going to be white or red. If it was incomplete, when we had the heterozygous condition, we can get pink, a mix of both phenotypes. Meanwhile, for codominance, both alleles are dominant. So in some cells it's going to be white, some it's going to be red. So you'll actually see splotches of white and red on the flower. So, what do you think we have here? Complete dominance, incomplete dominance, or codominance. We're talking about earlobes. Your earlobes can either be free or attached, one or the other. That would give us complete dominance. And there are a wide variety of other traits that work via complete dominance, such as the earlobes, being able to coil your tongue, a widow's peak, the folds of your eyelids, dimples, thin or normal amounts of hair, dark or light hair, the hitchhiker's thumb, and having your forefinger longer than your third finger. Now here, we've got white flowers, red flowers, and if we have a combination of both, we get pink. So we have a blending going on. What do you think? Would this be complete, incomplete, or codominance? That would be incomplete dominance. So neither trait is fully dominant over the other, resulting in a blending. Now here we go. What do you think? Eye color. Complete, incomplete, or codominance? This one might be a little tricky. Once again, this can be incomplete dominance. Though basically the darker colors tend to show a lot more strongly than lighter colors. So there might be lighter colors in there, but they'll be overshadowed by the darker colors. So as you can see, there can be a wide variety of different colors of eyes, and a wide variety of different combinations. That's the fun thing about eyes. No two eyes should look the same, well, between you and someone else. It's very unlikely. Which is this? Well, this is currently a polar bear hiding in a snowstorm. But check it out. We got ourselves a real groovy thing going on here, man. We got We got this like really cool flower. It's got like pink and it's got white, splotches everywhere. Like, what do you think? This would without a doubt be codominance. Both traits are showing, so we got pink and we've got white. So an incomplete dominance problem for you here. So go ahead and set this up like a normal Punnett square. In northeast Kansas, there is a creature known as the Wildcat. It comes in 3 colors, red, blue, and purple. This trait is controlled by a single gene with incomplete dominance. So a homozygous individual, big B, so homozygous dominant, would be blue. A homozygous recessive, little b, would be red. And a heterozygous individual, big B, would result in purple. So what I'd like you to do is construct a Punnett square here, showing us what would happen if you crossed the, what would be the genotypes and phenotypes if you crossed a blue wildcat with a red one. So go ahead, pause the video, set it up, and I'll be here when you get back. Now, once you've got that finished, I'd like you to attempt the next one. If we cross a purple cat and a red cat, is there any probability that they could have a blue offspring? Once again, go ahead, set it up, pause the video, answer it, and then I'll see you when you get back. So let's see how we did. If you went ahead and crossed a blue wildcat with a red one, you should have 100% of the offspring being big B, little b, resulting in purple. You've got the heterozygous condition. Now, if you cross a purple cat and a red cat, there is no way you'll have a blue offspring. The only possibilities that you'll get are red, little b, and purple, big B, little b. So let's do a little codominance problem then. So Longhorns can be white, CW, red, CR, or roan, which is kind of like white with red specks, which is resulting in CRCW. Roan longhorns have a mixture of both red and white hairs due to a codominant gene. So let's go ahead and do another Punnett square here. Let's cross a roan bull with a red female. So go ahead, do that, and get our genotypes and phenotypes for the cross, as well as the percentages. And once you have that taken care of, try and answer the question, can roan cows produce more roan cows? Pause the video here, go ahead, work on your Punnett squares, and then we'll go over the answers. Okay, so pause the video right about now. So the answers here, for our first cross, a roan bull with a red female, we should get 50% CRCW and 50% CRCR, giving us 50% roan and 50% red. Now, could roan cows produce? Well, ladies and gentlemen, that's about going to go ahead and close us down for today. I'd like to go ahead and thank you all for joining us. And in retrospect, let's think about what we talked about today. We talked about how you could do a dihybrid cross, which is dealing with more than one trait. And the ratio of those, if you do a heterozygous cross, is always going to be a ratio of 9 to 3 to 3 to 1. 9 showing both dominant traits, 3 showing one recessive, one dominant, 3 showing the other recessive dominant combination, and 1 always showing you the recessive for both traits. Now, we also talked about the different forms of dominance. We have complete dominance, where it's either one option or the other. We got incomplete dominance, where there is a blending between the two different phenotypes when we have a heterozygous individual. And we also talked about codominance, where neither one is fully dominant over the other, so both traits will show. So, once again, thank you all for joining us today, and until next time, I will see you in the next video. Until then, you all take care.